Okay, last video of the unit, unless for some reason this takes forever. It's going to be on cell cycle regulation. Hope you enjoyed the last two short ones because this one is not going to be that short. So, let's talk about the cell cycle and how it's regulated. Some cells are going to divide and need to divide more often than others. Not every cell is the same in how much it needs to divide, how often it needs to divide, even why it would need to divide. So, and also some cells won't ever divide. But what usually tells a cell that it is time and it should is a signal that it gets in the cytoplasm. In the meantime, we have these checkpoints, which we are going to talk about in detail in a bit. So, you can get stopped at any of the checkpoints, but G1 is a little bit of a special one because you can enter what's called a G0 phase, which is just non-dividing. So, the cells that never divide are considered to be out of the cell cycle and in G0 or G naught. Not G naughty, gross. Okay, let's continue. So these checkpoints. In eukaryotic cells, at least, we have three main checkpoints that a cell has to pass in order to actually go through a full turn of the cell cycle. So the first one is the G1, and this is the main checkpoint where most cells will either stop or go through. It's like the most critical one. This is where you're basically looking for, is there DNA damage that happened during mitosis? Because once we get rid of that nucleus, that DNA is kind of vulnerable. It's no longer protected, and so it can get damaged. And so G1 is checking for that. And if it is not damaged, then the cell is going to commit to divide when it gets its growth signal and it has the proper nutrients. G2 checkpoint. This is essentially checking that you replicated your DNA properly. That's going to happen in the S phase, if you remember. And so G2 checkpoint is checking, did we replicate our DNA properly? If not, it'll try and fix it. But if it can't be fixed, it'll send a signal to do apoptosis and kill the cell. Then our last checkpoint is the M checkpoint. This is actually in mitosis, about halfway through. It's right before anaphase, so right before the separation point. And this is checking to see if the chromosomes are properly aligned, the spindles are there properly. So basically checking, like, is everything lined up? Do we have all the chromosomes? Again, if not, it will get a signal to usually kill the cell if it can't repair it. But at the M checkpoint, it'll likely just kill the cell. So, talking about these in a bit more detail, the G1 checkpoint, again, is the most important one for us mammals, and you can go to G0 instead of actually going through. So this is the one place where you can enter this G0 phase. So the things it's looking for are, is the cell big enough? Has it grown, physically expanded its membrane, got more cytoplasm? Does it have enough resources? Does it have enough nutrients to make all the things it needs to and do all of its cell tasks? Is there enough room for it, or is it too crowded? Is there DNA damage, like we talked before? And then are there growth factors? So essentially checking, is there damage? And then do we have everything we need? Do we have all our supplies? If we have all our supplies and there's no damage, let's go ahead. And then some cells will just go to G0 and not divide. And it won't be because they have DNA damage or anything. It's just because that is what that cell wants to do. So G0 is where most of our cells live, actually. Most of our cells are not really in a state of active cell division. Some cells, like our blood cells, our skin cells, we have to replace constantly because they get used up so much and exposed to a lot of damage. So those will often have cells that make them, at least, that are going through the cell cycle. And then you have your liver, which has a lot of regenerative properties. If you've ever heard of like a liver transplant, you just take a piece of someone's liver and then you grow a whole new liver essentially inside of you. So that is because the liver is special and it can re-enter the cell cycle. And it has this ability because it's exposed to a lot of toxins. Our liver is what filters and cleans our toxins out. And so it's just naturally exposed to a lot of damage, and we have to replace the cells pretty often. G2 checkpoint. Again, just checking for DNA replication. Did that happen? If there's damage, it's going to try and repair it. If it can't repair it, it's going to die. This one's pretty simple and straightforward. The M checkpoint, again, is just making sure that the mitosis machinery is in place properly, and that everything is lined up. If you get something like one of the spindle fibers doesn't attach and you pull both chromatids to one side, you get what's called a non-disjunction event. We'll talk about those more in the next unit. But if the machinery is not there, it just gets a stop signal and it stops mitosis. Okay, so we've hinted at these things a little bit, but now we're going to talk about them in more detail. What actually is pushing ourselves through this cycle? What is telling us to divide? So there's two main regulatory proteins that have a role. Those are cyclins and cyclin-dependent kinases. So cyclin, word sounds like cycle, and it's because it's a protein involved in a cycle. And then if we look at the word cyclin-dependent kinases, or CDK, which I'm going to use CDK from now on, it's a kinase, so it's going to be a protein that phosphorylates something. It's going to add a phosphate to something, and it's dependent on cyclin. So essentially, it's a kinase for cyclin, is what we can think of it as. So those are some of our main proteins that are going to kind of control how we go through the cell cycle. Some other things that can control it, internal signals. If we don't get the spindle fibers, remember at that G2 checkpoint, that M checkpoint, then we stop. But it can also be external, so it can be growth factors that are just some chemical. It's released by other nearby cells. If it's a cancer cell, it might be releasing it to itself. But generally, a feature of cancer is that it does not really respond to these mechanisms well. So it's kind of ignoring the signals and just going through the cell cycle regardless of what the cell environment is telling it. So let's talk more about the cyclins and CDKs. So we track the concentrations of cyclins throughout the cell cycle, and it basically shows you this periodic sine wave-esque pattern where the cyclin concentration goes up and it builds, and then as mitosis is basically finishing, and in the process of mitosis, all of the cyclin gets degraded back to zero before it builds back up again and we divide again. So these are really important and very easy to mix up, but try to remember CDKs are always there. You always have the cyclin-dependent kinase. You always have CDKs. They're just not always active in doing things. The cyclins get made and destroyed depending on where they are in the cell cycle and other signals. So our body is, or not our body, the concentration of cyclin depends on the cell. The cell is making cyclin for itself. And so it kind of builds it up, and once it hits this critical point, we go through mitosis and then degrade it. So kind of the rate that we build up our cyclin is a way we can regulate how fast something is going to go through the cell cycle. If we build up our cyclin slowly, it's going to go slow. If we build it up fast, it's going to go as fast as it can. So the real thing that's happening is the CDK is binding to the cyclin, and the cyclin-dependent kinase then can phosphorylate something else, some other protein that's going to have a role in the cell cycle. So always have CDK. When we get enough cyclin, we activate a bunch of these proteins, and once these proteins get activated, the cell cycle continues, goes through, and we destroy the cyclin at the end. So you need both cyclin and the CDK for mitosis to happen. If you're missing one or the other, it will not occur. And the job of it is just to phosphorylate and activate a protein that is more directly involved in mitosis. So you could think of it, it's not really this way, but you could think of it as there's a protein that tells the body go through mitosis, and CDK cyclin complex is what activates that protein. It can only be activated when you have cyclin and CDK. We're going to talk more about these and work more with this because it is a bit confusing. Okay, let's talk a bit more about cancer specifically, which we are going to do an activity on this. So we have two main types of genes, at least that we classify them as, that control and regulate our cell cycle. The first are called proto-oncogenes, which is a really cool name. An oncogene means a cancer-causing gene, and a proto-oncogene basically means it's a gene that could become cancerous in the future. We all have them naturally, but if they become mutated, they tend to become cancerous cells. And they just generally code for proteins which promote the cell cycle. Cancer, again, is just basically going through the cell cycle too much, too fast when you're not supposed to. So the genes that are involved in telling the cell to go through the cell cycle tend to be involved with cancer. The other type of gene is the opposite. It is a tumor suppressor gene, and these are proteins that inhibit the cell cycle and or promote apoptosis. So you kind of have this balance, and in a healthy normal cell, it's a perfect balance where your proto-oncogenes are promoting your cell cycle, tumor suppressor genes are repressing it, and so only the correct cells are going through the cell cycle, and they're doing it at a good, normal, balanced rate. Cancer cells, though, break all of this. They are essentially just not going to respond to the inhibition. They don't need their own growth factors to grow and divide, so they don't need someone to, or another cell or something to say to divide. They can make their own growth factors and stimulate themselves with autocrine signaling. Sometimes they just express the signal, they just exhibit the response without the growth factor actually there. So you could think of just like whatever the growth factor is supposed to activate is just permanently activated in a cancer cell. Or the cell cycle control system itself is abnormal. So like your proto-oncogenes or your tumor suppressor genes, there's something wrong there. The diagram really just shows how cancer can kind of spread through the body. So it starts with a tumor from a single cancer cell, divides, and then as it divides, it starts to take up too much space, and so it starts to invade neighboring tissue, just fills in whatever space it can. And then once they get access to either the lymph or the blood vessels, your lymph is like your trash of your body in a lot of ways. It's just a bunch of junk that you're going to get rid of. And it's connected to your whole body. Same with your blood cells and blood vessels, So as soon as the cancer hits those, they will travel through the body and they can establish in other regions and form other tumors in other areas. This is when it is considered really, really bad. I think is it called stage 4? When it's undergone metastasis, meaning it has spread to a different location in the body. Once it hits the lymph nodes or your lymph vessels and your blood vessels, cancer becomes much harder to treat. When it's isolated in one area, it's much easier. So, to review, proto-oncogenes are like gas for the cell cycle. They tell the cell cycle to go forward, and our tumor suppressor genes are the brakes. In a normal cell, these are balanced so that we get a good constant rate. In cancer cells, there are two main types of mutations that can happen. You can mutate your proto-oncogenes so they become an oncogene, meaning you just get way too much gas pedal stuck, slammed down. And then your tumor suppressor genes can become inactivated or defective, meaning you don't have any brakes. Often cancer will be both. So you have all gas, no brake, and so your cells are just dividing over and over again taking up resources, space, all of that, which is very, very bad. And that is Unit 4. This one wasn't too long, 15 minutes. The last two were short. You can deal with it. We'll have our ASU final and a bunch of junk. Stay tuned. I really think the rest of the year has some of our coolest stuff to learn. So, looking forward to it. See you later. Bye.